Microarray studies and RNA-seq studies in Omics lands follow a universal pipeline. This allows our users to easily compare studies from different sources and to search for genes that may have similarly regulated genes under different conditions. Users can find inference reports from OncoGeo studies simply by typing the study name into the search bar at the top of the page. Here, let's type the study number GSE 78220 and notice that a drop-down menu will appear with all values containing this project number, including project name and subject ID. These will display a heat map of all molecular set gene signatures in this study or individual samples. Also in the drop-down are comparison views for project and individual comparison levels. Let's start with the project comparison view. This will create a volcano plot view for all inference reports for comparisons in this project, with fold change plotted on the x-axis and p-value on the y-axis. For this study, there are two types of comparisons performed using DE-seq, the first comparing partially versus completely responding to anti-PD-1 response. The second test compares samples with progressive disease or anti-PD-1 resistance versus complete response. Let's focus more on the comparison between anti-PD-1 resistant and completely responding to PD-1. Typing in the GSC ascension number again. Let's search for the test 2, which is the second test in this view. This will filter for just that volcano plot. As with all views in Array Studio, views in land are highly customizable. For this inference report, we can add cutoff lines using specify cutoff lines for both p-value and fold change. We can also change symbol properties to customize a number of features. For example, we can use continuous coloring and use the fold change from the inference report. As you can see, this instantly updates our volcano plot to color upregulation in red and downregulation in green. In addition, we can change the dots on this view using the size. Let's use fold change p-value from the inference report to change the size of the dots based on the significance. Here, higher significance will lead to a higher size or larger size in the dots, whereas lower significance will be much smaller. In symbol properties, we can also change the shape from squares to other shapes, such as circles or triangles. Finally, we can change what labels will be presented when points on this volcano plot are selected. Rather than gene symbol, let's select gene description to get an idea of what kind of gene it is. Simply selecting genes within this view will populate gene descriptions for each of the selected genes. Users interested in finding projects within OncoGeo that have common gene regulation as the genes in this inference report can select View and under Enrichment Analysis, Gene Set Analyses. In these analyses, genes from all comparisons are ranked based on p-value and at least a 1.25 linear fold change in any given experiment. If there are more than 100 genes, the top 100, ranked by p-value, are used to search for similar enrichment in other comparisons using a Wilcoxon test. This view is available as a plot or table for your convenience. Let's start with the plot view, where significance for gene set enrichment is plotted on the x-axis and comparisons are grouped on the y-axis based on disease category. In this example, there are two skin cancer comparisons with a high significance from this gene set. By left-clicking and drawing a box around these 2 points, we can view the details of these comparisons. Not surprisingly, these two studies are from the project we just examined. Let's filter this view to find studies of interest. First, let's switch to the table view of the gene set analyses. Here, we can use the metadata from the comparisons to filter for samples of interest. For example, since this study was done on skin cancer patients, let's use the metadata for disease category and select the box for skin cancer by right-clicking and checking this only. In addition to using the metadata from the comparisons, we can also use the results from the Wilcoxon test. For example, to find gene set enrichment that was significant, let's use the Wilcoxon test p-value and add a filter for less than 0.05. By applying these two filters, we have filtered the over 1,000 comparisons for 11 studies that have a gene set enrichment similar to the study of interest. The comparisons that will be left are shown in table format, ranked from top to bottom by p-value. As shown in the plot earlier, the top two comparisons correspond to the anti-PD-1 project discussed in these videos. Viewing in table format allows users to browse the metadata that's related to these comparisons. For example, scrolling to the right, we can find information regarding the response and treatment of the anti-PD-1 study. For one of the remaining samples, we can see one comparison is found that was related to MAPKi inhibition resistance. In this study, the authors examined the differential expression before and after MAPKi inhibition therapy, leading to clinical resistance to MAPKi inhibitors.